This is Dr. David Rufino Ramos. He's a research fellow at MGH and a postdoctoral fellow at Harvard. He's part of the Kleinsteiger lab and studies genome editing strategies. So thank you very much.
Thank you. Thank you for the nice introduction. First of all, I would like to thank the invitation to be here speaking at this conference. It has been a really pleasure to learn from all of you in a field that is slightly adjacent to mine. So, as it was mentioned, I came from a genome editing lab, and what I've been talking today is how can we apply genome editing approaches to correct pathogenic mutations. And the story that I'm gonna talk today is about how can we go from the genetic diagnose and draw on a genetic toolbox of genome editing therapeutic approaches to correct individual patient mutations. Going into the genome editing toolbox, so since the discovery of CRISPR-Cas9 enzymes, the field has been evolved a lot, and we start using nucleases that are enzymes that are able to perform double-strand breaks in the DNA, and they are quite useful to do knockouts, as was mentioned here before. Then a group in the Broad Institute here in Boston, led by David Liu, developed base editors, and this is very precise tools that are able to modify one specific base to convert to another base. Later on, 2 years later, the same group create prime editors. Now, a genome editing tool that is able to write new sequences of the DNA. And what is remarkable is that now these genome editing tools have been used in the clinic, and they have been proven to correct patients' mutations. We also have some emerging new tools, such as click editors, that basically can offer the same ability as prime editors, so can write new portions of sequences, of new sequence in the genome. And we also have large gene insertions technologies that are now being developed and is where the field is moving towards, and this is the frontier of the genome editing technologies. So I'm going to spend the next couple of minutes talking about base editors, and if I have time at the end, I'll go briefly to the large gene insertion. So what are base editors? Base editors are a genome editing tool that is composed by 3 elements. The first one is a nickase Cas9 that is associated with a guide RNA, and together, this guide RNA drives the nickase Cas9 to a specific target site into the DNA. When the Cas9 binds to the DNA, it unwinds the DNA, and it exposes one of the strands to a deaminase. And the deaminase, the function of the deaminase is actually converting one base to another. For example, it can convert adenine, here highlighted as an A, to a guanine. What is remarkable about these base editors is that they are modular, which means that these 3 components can be engineered separately and can be combined in order to maximize the editing efficiency and the safety of this tool. Of course, one of the biggest challenges of the base editors is actually a property that is inherited to the Cas9 enzyme, which is the PAM. The PAM is a motif, a sequence of the DNA that the Cas9 needs to recognize in order to bind. In the case of the wild-type Cas9, this PAM is 2 guanines, 2 Gs, as highlighted in blue. And in the case of base editors, this is actually really important because we need to place the deaminase several bases apart from the PAM in a region that we call the edit window, as you can appreciate here. So the mutation that we want to correct needs to be roughly 16 bases apart from the PAM, and the deaminase needs to act precisely on this mutation that is here highlighted as an A. within this edit window, that is actually the region where the deaminase can act, if we have other bases that can be deaminated, it's going to lead to bystander edits, which are unintended edits that we do not want to have when we are performing corrections of the genes. One of the drawbacks of this is that when we are using wild-type Cas9, we are limited to the PAMs that exist in the region nearby of the gene that we want to correct. This turns out to be a huge opportunity for groups like us and others to perform engineering campaigns in order to create enzymes that can broaden the accessibility to the genome. And a couple of years ago, our group created SPRY. SPRY is an enzyme that maximizes the genome accessibility. So now we have an enzyme that can basically access to all the genome, without any PAM restrictions. So this enzyme is near PAMless. Then, much more recent, we start thinking, how could we even create better tools? And we thought, how if we could somehow combine the specificity of a PAM, of the wild-type Cas9, with the broad range accessibility of SPRY. And very recently, we create a machine learning model that is able to nominate a bespoke enzyme to specifically target a sequence. So instead of having one enzyme that can access to all the genome, we now have one enzyme for each PAM that we want to access. And this turns out to be so efficient that it's already in patients. One of the stories that I'm going to tell you today is about the story of the baby KJ that came out in the news a couple of months ago. And this baby was the first patient treated with a bespoke editor. This study was led by Kiran Musunuru and Rebecca Aaron-Nicholas in SHOP. And basically, this story is that baby KJ was born with a single point mutation in the CPS1 gene that caused a deficiency in the urea cycle metabolism, and basically the levels of ammonia of the baby KJ were extremely high in the blood. I'm not going to get into the details of how we correct this mutation, but what I'm going to highlight here is that from the first diagnosis until the correction, the first dose of the patient, it was less than 7 months, which is a remarkable achievement when we consider that we need to develop an editor, we need to involve the regulatory authorities, and we need to also have the infrastructure of the companies to create this therapeutic tool in order to give it to a patient. As a consequence of this, the baby was then dosed more 2 times, so in total 3 times. The level of ammonia were reduced, and the baby was able to leave the hospital and to be with his family. This is a study published a couple of months ago, and it's a very inspiring story. And if you have some time during this next couple of days, I would invite you to watch a YouTube video that is about 10 minutes about this story that is very inspiring, very motivational, and very remarkable. But why am I telling you this today? How can we make the baby KJ story a reality for more patients, a reality for patients, for ALD patients, for the ALD community? And as we've been talking today, the ALD disease is caused by multiple mutations, by different mutations in the ABCD1 gene. And there's a lot of these mutations that are single base pair mutations, which means that they can be treated by base editors, by bespoke base editors. So how we can generate, how can we leverage this toolbox that we generate in our lab in order to start correcting these mutations? So as a standard point, we start with a Cas9 that has accessibility to a very specific PAM. We brought the accessibility of these enzymes either to the entire genome or to nominate bespoke editors. And now we basically have a toolbox of enzymes that we can combine, and we can do multiple tests in order to precisely identify the best editor candidate to treat each mutation. So we kind of solve some of the three big challenges in the field of genome editing, that is targetability, activity, and safety. So the base editors were modular, as I told before. So they can be, we can engineer the aminases, we can combine multiple Cas9s, and we can even test multiple guide RNAs. So basically, we can now are able to tile all the combinations in each mutation. The next question is, how can we perform this in a disease like ALD, where we have lots of mutations to test? And the conventional approaches are usually generating an endogenous cell line that takes a couple of weeks to make, and we go from a single colony to expand these cells, and then we can start testing all the candidates. This is feasible for one mutation or two mutations, but if you want to test multiple of these mutations, it gets kind of very unreasonable in terms of workload and in terms of time frameness. So what we and others have been proposing is to create a reporter cell line model where we can attach together all the mutations that we want to treat. This can be done within the same mutations within the same gene or across multiple genes. And these cell lines we can create in roughly 2 weeks, and then we are able to test all the genome editor candidates. Because we want to test multiple mutations at the same time, we are even able to leverage multiplexing capacities of our genome editors in order to generate a compiling piece of data in a very short time frame. And how does this work? We can generate these reporter cell lines with lentivirus or with transposases or integrases, and basically we can compile all the mutations that we want to treat. We can even, in case of ABCD1, we can even treat mutations that are very close to each other and basically isolate each gene context. So we can screen for all of them, and then we can tile all the guide RNAs, all the possibilities of combinations to use base editors. And here I bring one example of one target site for the ABCD1, and on top you can see that just to remind you that what we want to target, the place where we want to target, we call the on-target editing. And if you look to the heat maps, on the dark color, we have the on-target editing that is roughly above 60% in reporter cell lines. Here we test 2 different base editors and 6 guides for each editor. And if you look down to the on-target, we can look to the bystanders, so these unwanted edits that we do not want to have to correct the mutation. And the profile of one editor comparing to the other edits is completely different. By doing this, we can precisely choose what would be the best guide RNA editor candidates to move forward for safety studies. This is one site. This is also a representation of one site. Imagine that with the same amount of work, we can now generate data for 20 sites, for other 19 sites. This allows us to, in a very reduced time frame, to nominate which are the best bespoke editors to treat each mutation. And then we move forward to investigate even further, to use bespoke editors to try to identify what is the best on-target editing with minimal bystanders. And in this example, we can see that candidate 9 and 10, despite not be the one that have more precise editing, is actually the one that offers a better ratio between precise editing and bystanders. So I hope I can convince you that using base editing technologies offer a good solution to treat single base pair mutations within the patients. However, in the ALD disease, there are lots of mutations that are not single base pairs, are, for example, deletions. And for that, we need to use other genome editing technologies that allow to write portions of the DNA. However, I'm not going to focus on this today, but I'm going to tell you a different story. A story about how can we leverage the use of large insertions to provide a solution for all mutations in the ALD context. And for that, what we can do is basically we can try similar to what was done here with the lentivirus reporter cell lines, but now we can try to insert or to replace part of the gene or even the whole gene into an early intron of the ABCD1 gene. And we did this in our lab. So this is basically a mechanism, two-steps mechanism. In the first part of the mechanism, we need to place a landing site that is here highlighted as a HATV. And for that, we can use genome writing tools, such as prime editing or click editing. Then, this landing site is an exogenous sequence to the genome, and basically, it's going to recruit a recombinase that drags on the new gene that we want to insert. If a recombinase event happens, the gene is inserted, usually in an early intron of the ABCD1 gene, and then this gene is going to be expressed under the endogenous promoter of the gene. We tried this in the ABCD1 context. So we start using ATV reporter cell lines, so the landing path was already pre-installed, and when we deliver the recombinase and DNA containing the ABCD1 gene, exon 2 to exon 10, if a functional insertion is happening, we would start having a production of a new protein while we were turning off the old gene that is there. So to facilitate the measurements in the laboratory, we associate a protein, we associate a reporter system at the end of the protein that allows to measure light if this protein is existent. And very remarkably, we were able to see this protein being produced in cells, and this was very inspiring for us. And that led us to move forward to engineering even more this system. Despite this system being very promising because it can offer a therapeutic agnostic system to correct every mutation in the ALD population, still is very preliminary, and basically we are now engineering each component of the system, which I mean engineering the way to efficiently install the landing path, engineering the recombinase, and engineering the way we can deliver this donor DNA in order to make this therapeutic tool available to be used out of these reporter cell line models and to be used in other cells and hopefully be used in the clinic later on. So, as conclusion, I would like to tell you that we have base editors that are clinically actionable now, that can be used now to correct mutations, single base pair mutations. They have been used in the clinic, and hopefully what we want to do is to generate a compiling piece of data that we can deliver to the authorities, and basically that we can correct not only one mutation, but this data that we can correct multiple single base pair mutations within the ALD gene. This is base editors. They are clinical actionable now. We are also working at the frontier level where we want to actually insert the gene to be able to correct all the mutations in the ABCD1 and in other diseases. As a highlight, I would like to thank to my lab, to my mentor, Ben Kleinsteiber, and also to all my colleagues to make this lab environment a very great place to work. Also to all the collaborators, Florian, and Yeda. They have been amazing working with us in the ABCD1 project. And I also would like to highlight two amazing students that put a daily effort every day to move this project forward, Lorenzo and Dalia. They are very excited to be here, and they also have posters to share some of our science also with you. And thank you.